Now that we have understood the light reaction, both pathways, non-cyclic and cyclic photophosphorylation, let us see the second phase of photosynthesis, that is dark reaction. And as we have discussed that this reaction is light independent reaction, that means it can take place in presence of light as well as in absence of light. This reaction is also known as biosynthetic pathway. And the reason why it is known as biosynthetic pathway is that by using the ATP and reducing power which is synthesized during light reaction, in this process sugar synthesis, that is carbohydrate synthesis, will take place. The entire process of this dark reaction was explained by Melvin Calvin, and he used carbon 14, that is radioactive carbon. C14 was used to trace the pathway. Now the experiment which he performed was he used green alga, exposed them to sunlight and took certain green alga cells and killed them in hot methanol, and after a few minutes when he would kill those cells, he examined the products which were obtained in those green alga. That's how he kept tracing the path of this carbon 14, and whichever compound had this carbon 14, he was able to interpret that these compounds were formed first, then next, and so on. That's how we got the entire cycle. The first compound which is formed is a 3-carbon compound called phosphoglycerate or phosphoglyceric acid. Phosphoglycerate or phosphoglyceric acid. So now, as the first stable compound which was found out was a 3-carbon compound, what they anticipated was that when carbon dioxide, which is 1 carbon compound, is taken in, probably it is accepted by a 2-carbon compound. And that's how this 3-carbon compound, which was the first compound, was formed. But later on they found out that the compound which accepts carbon dioxide is not a 2-carbon compound. It is actually a 5-carbon compound. And this compound was found to be RUBP, that is rubulose biphosphate. It is a ketone sugar. And it is a 5-carbon compound. So initially when the first compound was isolated and it was detected to be a 3-carbon compound, what was an easy guess was that 1 carbon containing CO2 must be accepted by a 2-carbon compound, and that's how this 3-carbon compound is synthesized. But that was not true. They found out that the carbon dioxide acceptor is a 5-carbon compound. Another very important thing which we must know in this process is that when carbon dioxide gets added or is accepted by this RUBP, then the process is known as carboxylation. And for this process, an enzyme helps. The name of the enzyme is Rubisco. Now what exactly this Rubisco means? It means rubulose, that is for rubis. For rubulose they have taken rubis. carboxylase, oxygenase. That means this Rubisco, it can act as a carboxylase also and oxygenase also. But the nature of this enzyme is decided by the external factors like temperature and concentration of carbon dioxide and oxygen. So in certain conditions it is going to act as carboxylase, and if those conditions change it is going to act as oxygenase, and that is why it can work as carboxylase as well as oxygenase. Carboxylase means it is going to help in addition of carbon dioxide, and oxygenase means it is going to help in addition of oxygen. We will take this up a little later again, but this is the most important enzyme which is required for this process. And as we have seen the name of the scientist, this pathway, that is dark reaction, is also known as Kelvin cycle, and it is completed in 3 steps. So let us draw those steps of Kelvin cycle now. So the 3 steps of Kelvin cycle. Kelvin cycle, 3 steps. First is carboxylation. The second step is reduction. And third step is regeneration of RUBP. For our convenience, to understand the entire process clearly, we are going to start with 3 carbon dioxide molecules accepted by RUBP. And by the end of the cycle, we will understand why we have started with 3. But in nature, when plants synthesize glucose, they start with 1 carbon dioxide. That means every time only 1 carbon containing molecule, that is CO2, is accepted. So this is carbon dioxide molecule which is accepted by RUBP, that is rubulose biphosphate. We have to understand the number of molecules which we are starting with, and we will keep a track of number of carbon atoms. We will start with 3 carbon dioxide molecules. So 1 carbon from each carbon dioxide, so there are 3 carbons here. RUBP is a 5 carbon compound, and 3 RUBPs are going to accept 3 CO2s. This is a 5 carbon compound. That means the total number of carbons which are coming from here are 15C. The enzyme which is going to help in this process is Rubisco, that is RUBP carboxylase oxygenase, and here it is acting as carboxylase. It is helping in addition of carbon dioxide to RUBP, so acting as carboxylase. 15 carbons from here and 3 carbons from here. We know that the first stable compound which Kelvin isolated was phosphoglycerate, which is also written as 3-PGA. PGA is the short form for phosphoglycerate or phosphoglyceric acid, and 3 is the number of carbon on which this phosphate is attached. This phosphoglycerate is a 3 carbon compound. 6 molecules of this 3-PGA will be synthesized. 6 molecules, each having 3 carbons. So there would be 18 carbons in all. 15 were coming from RUBP, 3 are coming from carbon dioxide, total were 18, and here we are able to have those 18. Now, this phosphoglycerate, where the phosphorus or the phosphate is on the 3rd carbon, it undergoes phosphorylation. There are 6 PGAs and there would be 6 ATPs required to give their phosphates here. So we will get 6 ADP, and the molecule which we would get here is 1,3-diphosphates. phosphoglycerate. It is the same phosphoglycerate, that is the acid, but it is diphosphoglycerate. That means originally it was having only one phosphorus. The second phosphorus has come from the ATP and now it has two phosphates. One at carbon number 1 and the second at carbon number 3. And phosphoglycerate is again a 3 carbon compound. So here we will get 6 molecules. 6 molecules of this 3 carbon compound. Each molecule is having 3 carbons and there are 6 molecules. So again there are 18 carbons which we are able to trace here. This diphosphoglycerate, it undergoes reduction. That means now we are coming to the second step, reduction. And for reduction there has to be some agent which is going to reduce it. So the reducing power which was synthesized during light reaction, that is NADPH2, will reduce the acid into aldehyde and we will get NADP, and these inorganic phosphates, 6 of inorganic phosphates will be released. So 6 of these NADPH2 will be required. 6 NADP will be produced here, and here there are 2 phosphates with each molecule and there are 6 molecules. So 6 phosphates are lost as in the form of inorganic phosphates. 6 phosphates are still there and the molecule which is synthesized is PGAL, phosphoglyceraldehyde. So from acid now we have an aldehyde and it is still having 1 phosphorus. This phosphoglyceraldehyde is also a 3 carbon compound. So 6 molecules of phosphoglyceraldehyde. That means again the carbon numbers are 18 because each molecule is a 3 carbon compound and we are getting 6 molecules of it. So 18 carbons. We are still able to keep a track of it. Now out of these 6 molecules, 5 molecules will help in the next process and 1 would get separated. That means 5 PGAL molecules will come here and 1 PGAL molecule will get separated. And this, let me write it as 5 molecules and this as 1 molecule. So this 1 molecule is going to help in glucose synthesis. That means so far there are 3 carbons which have been separated or have been taken out from this cycle. Let us check the carbon numbers. PGAL is a 3 carbon compound. So these 3 carbons, because there is only 1 molecule, come out of the cycle. PGAL, which is a 3 carbon compound, and 5 molecules are getting separated for the next part. So 3 from here and 5 molecules. So there are 15 carbons in this case. That means each phosphoglyceraldehyde is having 1 phosphate with it. And there are 5 molecules. So if we just sum up, there are total 15 carbons and there are 5 phosphates with it. So now in the next step, 2 inorganic phosphates are released. How many phosphates would remain after 5 phosphates have been removed? Let me just explain this one more time. Phosphoglyceraldehyde is a 3 carbon compound. So if we have to count the carbon numbers, 3 carbon compounds, 5 molecules. So total carbons are 15. Now from here, 2 phosphates are lost. How many phosphates were there total? Each PGAL was having 1 phosphate. And there were 5 molecules. So that means there were total 5 phosphates. 2 are given out in the form of inorganic phosphates. So now what remains? 2 phosphates are lost. So total 15 carbons are remaining and only 3 phosphates are remaining. So we get here Ru, rubulose, 5 phosphate. Rubulose 5-phosphate is a 5-carbon compound and there are 3 molecules of this rubulose 5-phosphate are synthesized. Let us keep a track. 3 molecules of 5-carbon compound. That means total carbons are 15. Here there were 5 phosphates. 2 got lost in the form of inorganic phosphate, so 3 remain. Each molecule is having only 1 phosphate, that is at position 5 or at carbon 5. And now it undergoes phosphorylation. So there are 3 molecules here, 3 here. These are rubulose 5-phosphate, or we can say rubulose monophosphate. It has to change into rubulose biphosphate. So here 3 ATPs will be required and 3 ADP will be formed, and those 3 phosphates will go here, and that is how this monophosphate will change into biphosphate. Again, how many carbons are here? 15. And the RUBP, that is biphosphate, which is regenerated, is also having 5 carbons. So in first step, that is carboxylation, what was happening was carbon dioxide was accepted by RUBP, rubulose biphosphate. The first stable compound which was formed was a 3-carbon compound, and this is the reason why this cycle is also given another name. It is called C3 cycle, because the first compound, stable compound which is formed, is a 3-carbon compound. This is a 3-carbon compound, and because of this the name C3 cycle has been given. So this is step number 1, that is carboxylation. The next step is acid getting reduced into aldehyde. For this, ATPs are also required and NADPH2, that is reducing power, is also required. These two are synthesized during light reaction. 6 molecules, because we have started with 3, 6 molecules of the aldehyde are synthesized. 1 molecule comes out of the cycle, that means 3 carbons get separated. And 5 molecules would help in regeneration of RUBP. Now the reason why we started with 3, because from 6 molecules, separating 1 molecule and 5 looks easier. If we would have started with 1 carbon, we would have been required to separate fractions, and then the calculation part becomes difficult. So this is for our understanding, but in real sense, that is in plants, there are 6 Kelvin cycles taking place for synthesis of 1 glucose molecule. 6 Kelvin cycles are required. This we have to keep in mind. Only reason why we are starting with 3 carbon dioxides, or we could have even started with 6 carbon dioxides. This is only to understand the process in detail and to avoid that separation of molecule. Imagine if only 1 PgAl was synthesized. So we would have said that 1 fraction goes in one part and 5 fractions go in the same pathway. So to avoid that fraction confusion, we start with 3. Suppose this whole cycle repeats one more time. 1 more PgAl will come out. So 1 PgAl is giving us 3 carbons. Second will give us 3 more carbons. That means 6 carbons will get separated, and that will help in formation of glucose. Now let us count what is required for synthesis of 1 glucose molecules. How many ATPs are required? We are starting with 3, so we will count it 2 times. So in this case, 6 ATPs are required here, 6 ATPs here, and 3 ATPs are needed here. 3 ATPs are needed here. That means we need 9 when we are starting with 3 carbons. But ultimately, to synthesize glucose molecule, we'll have to repeat this reaction one more time. So 3 carbons, 1 PgAl separated, one more 3 carbon, another PgAl will get separated. That means 9 and 9. So 18 ATPs are required to synthesize one glucose molecule through C3 cycle. The second thing which is required is this NADPH2. So when we start with 3 CO2, 6 NADPH2 are required. When we start with 6 CO2, double of this. So 18 ATPs are required and 12 NADPH2 would be required. So for one molecule of glucose synthesis, one molecule of glucose synthesis, what is required is 18 ATPs and 12 NADPH2. Again, in plants, every time only one carbon dioxide is accepted by one RUBP. We have added this number to make this cycle easy to understand, but this thing is very important, that there are 6 Kelvin cycles which take place for synthesis of one glucose molecule, and there are 18 ATPs required and 12 NADPH2 required when one glucose molecule gets synthesized. So the 3 reactions, that is carboxylation, addition of carbon dioxide, then reduction, the acid which is synthesized gets reduced into the aldehyde, and third, regeneration of RUBP. So here RUBP gets regenerated so that it can participate in this reaction. One more quick recap. The reason it is known as Kelvin cycle, because the entire path was traced by Melvin Kelvin, and he used radioactive carbon, that is C14, to trace this path. It is also known as C3 cycle because the first stable compound which is synthesized is a 3 carbon compound. So it is known as C3 cycle, and it is also known as dark reaction because it is not dependent on light directly, but indirectly it is dependent, as ATP and NADPH2 which are required for this are synthesized in the light phase. But this pathway can continue in light or in absence of light also. So this term dark reaction. It is also called biosynthetic pathway because we are synthesizing carbohydrate or glucose through this process. So this is dark reaction. Now in the beginning of this particular section we said that this Rubisco can act as carboxylase as well as oxygenase. So now we will take up the conditions in which the behavior of this Rubisco changes, and then we will see how that changed Rubisco brings about changes in the process.